right, so thank you all. Welcome. This is the first of our RaHP VEC summer seminars for this year. We'll have these every 2 weeks at this time, and they'll all be recorded, so they'll all be posted online, hopefully tomorrow or sometime this weekend, and then distributed around. Probably not for you guys that are on the call today to watch it again, but for those of you who can't join us to watch it. And all right, so our first speaker today is Dr. Chris Roundy. He is the state public health entomologist for Colorado, and he works for the Colorado Department of Public Health and Environment, CDPHE. His work focuses on surveillance, prevention, and control of disease vectors throughout the state, including mosquitoes, ticks, fleas, and kissing bugs. And Dr. Roundy also serves as a subject matter expert as needed for county public health officials, local mosquito control districts, and academic partners within Colorado, as well as regionally through his role as an investigator within the Rockies and High Plains Vector-Borne Disease Training and Evaluation Center, which hopefully we're all familiar with here. He has led an expansion of the state's tick surveillance program, which I am sure he's going to talk about today, as well as helped counties throughout Colorado grow their mosquito and West Nile virus surveillance programs, as well as assisted with the response to the emergence of an invasive mosquito species, Aedes aegypti, which some of you may be familiar with in Western Colorado. Dr. Roundy obtained his PhD in biomedical sciences from the University of Texas Medical Branch, studying vector virus interaction between Zika virus and various mosquito species, and obtained his Master's of Science in Public Health, studying tropical medicine at Tulane University. All of these things are things that he is about to tell you about, I think, and is happy to take questions at the end about as well. So without further ado, take it away, Chris.
Thank you for quite the introduction, Tim. Get my screen sharing. All right. So while I'm presenting, actually I've met one or two of you. I've not met many of you, and a lot of the things I'll be talking about today are, pieces of projects that you're all involved in. So while I'm presenting, if you wouldn't mind putting your name in the chat, the agency that you're working with this summer, and then just, a really quick description of the project or topic that you're working on. So as Tim mentioned, I'm Chris. I work for CDPHE as the state medical entomologist. So where does the state medical entomologist live? I'm within CDPHE's Zoonoses in One Health program. This is the only picture I could find of my team. This is from Halloween last year. And as you can see, I think we're a pretty fun bunch. We all dressed up as either animals that we study, vectors, reservoir species, things like that. There are a couple of people missing from this photo, but I think it shows the spread, the breadth of things that we work on zoonoses and One Health, including bird flu, New World screwworm, rabies virus, hantavirus, and then, of course, the vector-borne diseases that fall under my umbrella. As the CDP medical entomologist, what my role really is to support vector surveillance and control throughout Colorado. That means, as Tim mentioned, really serving as a subject matter expert for a number of partners throughout the state. Anything on vectors or the pathogens that they transmit are things that I am expected to have some pretty deep knowledge on. This often involves training local health departments as well as other partners. If a county wants to start doing mosquito surveillance and they need some help on, what trap types to use, how to set them up, how to identify the mosquitoes that they collect, or similarly with ticks. I do a lot of work making sure that we are providing kind of good and accurate outreach and education to the public as well, which I'll touch on. I'm also covering identifying opportunities for funding for projects that we can participate in around the state. And actually, RaHP VEC is a great example of this. A few years ago, one of the professors at CSU, Brian Foy, who many of you may know, as well as my supervisor, Natalie Marzek, who's the supervisor for the Zoonoses in One Health team, independently reached out to me for some input on writing an application to be this new regional training and evaluation center. And I was able to kind of connect the dots between those two groups and put together a comprehensive joint application, which turned out very well for us. And I've been very pleased with this group so far. I also write, we aren't as dependent upon grants as many in academia, but grants are how we do get some of our funding, especially for like new surveillance methods or new approaches that we want to take. Those often start as funded by grants. One of the most common grants that we in state level public health pursue is the ELC grant, or the Epidemiology and Laboratory Capacity Grant. I also represent CDPHE in both regional and national meetings, as well as groups. And then lastly, for all of the data that we collect on mosquitoes, West Nile virus testing, tick surveillance, other things that I'll touch on in this, I do the data management and the analyses for that information. So you'll notice throughout this meeting, I'm using both medical entomology and public health entomology. And in my mind, there's not a real significant difference. Medical entomology is a broader term. Public health entomology lives within medical entomology. And I found that using the term public health entomology more accurately describes my role and is less confusing for people. In my role, I'm primarily focused on the bigger picture, large trends, vectors. Very rarely do I consult on individuals in any kind of medical capacity. So shifting from medical entomology to public health entomology has made it more clear what my role is. So during this presentation, I want to talk about some of the things that I've been able to do in this role. I want to emphasize that these are just highlights, kind of new and exciting things. I've been in this role for about 4.5 years now, and every year is a new challenge or opportunity, so it has not gotten boring. The vast majority of what I do is West Nile virus and mosquito surveillance throughout the state. A couple of years ago, as we'll get into, Aedes aegypti showed up in Grand Junction. Just last year, we also had the re-emergence of St. Louis encephalitis virus. I have significantly expanded our tick surveillance here in Colorado through both passive and active surveillance programs. Lately, you have probably heard, we've got New World screwworm re-emerging in the US as well. And then lastly, I'll talk about some of my work with the media. Mosquito surveillance in Colorado, as I mentioned, it's kind of my bread and butter. It is the most core piece of my role, and it was the first thing that I really had to learn and focus on when I started here. One of the tricks about mosquito surveillance in Colorado and in many states is it does not look uniform all throughout the state. There are many different counties and different partners doing different things. In Colorado, we have about a dozen counties that collect mosquitoes, identify them, and send them into our state public health lab for West Nile virus testing. In addition to that, there are, I believe, 4 mosquito abatement districts. They often work very closely with their local public health in the counties. We also have counties that contract that work out to companies. The most common in Colorado is Vector Disease Control International, or VDCI. Each of these approaches looks a little bit different. And it was one of my first real challenges when I started in this role to get a better understanding of what all of these pieces look like, where are opportunities to connect them, bring everyone to the same table, and then opportunities for expansion from there. So while year to year, we do have West Nile virus here in Colorado, some years are much worse than others. In a routine year, I am supporting counties, VDCI, academic partners, making sure that we have testing flowing, that the data is being accurately collected and managed, so providing training where needed, and then hopefully the season goes smoothly. That was not the case in 2023. So in 2023, Colorado had our second worst West Nile virus year on record in terms of human cases. The only year worse than yet this was 2003, which is the first year that we had the virus in Colorado. So we did not have a lot of infrastructure in place. We had no underlying immunity in our human or our bird populations. That was a really bad year. 2023 was almost as bad. So we can see on this graph, the y-axis is the number of human cases. The x-axis is epi weeks throughout the year. The bars along the bottom are the 5-year average prior to 2023. And then that line shows our cases that year. And it kind of speaks for itself. This season started early. We knew early on that this was going to be a bad season. Part of this was just how wet it was. If anyone lived here in the winter leading into 2023, great winter, excellent snowpack, great snowboarding all year. This was followed by a wet spring, all of that water. We just had a crazy number of mosquitoes. Um, we had an early detection, ramped up really quickly. And then thankfully, after it peaked, it dropped very quickly as well. But you can see we had over 630 confirmed cases. 50 of those individuals unfortunately died. 315 of them had neurologic West Nile virus, and 380 required hospitalization. So during this year, my job looked fairly different. I was doing a lot of outreach, making sure that local public health that was pushing the message made sure that they had the capacity to warn people about this, to push messaging about protecting yourselves, avoiding mosquitoes, wearing insect repellents, also securing emergency funding so that we could do more mosquito surveillance because the surveillance we were doing was just saturated. We weren't hitting the upper limit of where West Nile was until we expanded testing this year. Kept me very busy. And then in 2024, it was a fairly quiet West Nile season. It was nice. And then late in the year, I got news from Grand River Mosquito Control District out in Grand Junction that they had confirmed Aedes aegypti in their county. I hope that we're all familiar with Aedes aegypti. For those that are not, it is primarily a tropical mosquito, tropical, subtropical. Globally, these are a very important mosquito for public health. They transmit Zika, yellow fever, dengue, chikungunya. And while they had been seen in the US, it was primarily in the South and along the coasts. But leading up to this, there had been a growing presence in Southern California and in Vegas and then in Southern Utah, and they were just creeping their way a little bit further east until they showed up here. This is a map of specifically where in Grand Junction these mosquitoes were found, a neighborhood called Orchard Mesa. This is just south of Grand Junction proper across the river for those that are familiar. So looking backwards, in 2019, a single Aedes aegypti was found in a trap in Fruta. A few years later, 2 adults were found. This was 1 adult in 2 separate traps at different times during the surveillance season. Following both of these detections, Grand River increased surveillance in the area, put up more traps, identified more mosquitoes. They didn't find any other aegypti. The first time, they thought maybe this was someone that had just traveled from southern Utah where they had a larger aegypti problem, and they just happened to end up in one of our traps. In 2023, when you see it again and you see it twice, much harder to explain that away. So in 2024, Grand River created an aegypti-specific surveillance team, looked all around Grand Junction and throughout Mesa County. And as soon as they started looking, they did find it specifically in the Orchard Mesa area. So about 450 eggs, almost 800 adults collected that year. We were able to send samples to Colorado State University for some insecticide resistance testing, which found that unfortunately they were highly resistant to both deltamethrin and permethrin. This was not too surprising because the aegypti populations in the West have been found to be fairly resistant to a lot of the common insecticides used. But it was good to confirm that so that Grand River could make data-driven decisions on which insecticides to use for these populations. Um, Last year, they continued that surveillance, collected far fewer Aegypti, but it's important that they were still very steadily present. One of the questions we had in 2014 was, can Aegypti even overwinter in Colorado? When we looked at the literature, there wasn't much information on like the lower temperature bound of Aegypti. That's because in many places where they're found, it doesn't really matter. In Texas and Southern California, it's not getting cold enough to kill off Aegypti. So the fact that they showed up again in number in 2025 answered our question. In 2025, we were also able to submit some samples for genetic sequencing, and this found that they were closely associated with populations in California and Arizona. While that doesn't answer the question of how they got to Western Colorado, that does confirm that this wasn't a novel introduction from Central or South America. Currently, those Aegypti are localized to about 230 acres centered around Orchard Mesa. I was just talking with Tim, who is the director of Grand River Mosquito Control District. Tim Moore, not Burton. We get that confusion on calls all the time. Um, and he said that they are doing much more expansive surveillance this year. For the last couple of years, when they found them in Orchard Mesa, they really focused in on Orchard Mesa, trying to understand the problem, get things under control if they could. This year, to make sure that they aren't spreading elsewhere in Mesa County, they are using, I believe he said, over 100 traps around the area just to confirm that Aegypti isn't spreading elsewhere undetected. Last year, as we were conducting our normal West Nile virus surveillance, we also had some detections of St. Louis encephalitis virus. A little bit of history on St. Louis encephalitis virus for those that aren't familiar. St. Louis encephalitis virus used to be the common mosquito-borne virus in the US up until late 90s, early 2000s. What happened in late 90s, early 2000s? West Nile virus was introduced. Something about West Nile virus's introduction pushed out St. Louis encephalitis virus. There's been lots of hypotheses about viral competition in either birds or mosquitoes. I've not seen convincing data on exactly how that happened. But however it did, the St. Louis encephalitis virus was pushed out of the US. There were still some cases, but far fewer than there had been previously. It continued to circulate in parts of South America, especially in Argentina. In 2014, the virus was reintroduced, an Argentinian strain, into California. And something about that strain of the virus, it was able to get another foothold. From there, it slowly spread through the West. One of the big red flags for us was that it started to be more commonly detected in mosquitoes in Utah. And then last year, we detected it here. So it's important to note that during this time, the state public health lab was not testing for St. Louis encephalitis virus. However, we had other partners around the state that kind of served as our sentinels. They had a better capacity or an academic interest in testing for SLEV, and they're the ones that detected it. So in Larimer and Weld County, these detections were done through testing in Dr. Greg Ebel's lab at CSU. And then in Durango, that was detected by a mosquito abatement district, Animas Mosquito Control, who, instead of just testing for West Nile, were testing on an arbovirus panel that included SLEV. Based on these detections, and increased activity in Utah, California, other parts of the West. This year, the state public health lab will be testing all mosquitoes that come through us for West Nile virus testing. We'll be testing them for SLEV as well. I'm really curious to see what we find. So shifting from mosquitoes to ticks, prior to me starting with CDPHE, there wasn't someone who was a dedicated medical entomologist. That was covered by someone who was more of a veterinary epidemiologist. As such, there was limited capacity in what she was able to do, opportunities she was able to take advantage of. She had a passive tick surveillance program primarily aimed towards veterinarians. So if veterinarians found a tick, they could send it in for identification. I took that program and really blew it up into a statewide community science program. So if anyone found a tick, they can send it in for identification. This gave us a much broader baseline of data on what ticks were being found where. Um, those ticks come straight to my desk in an envelope to make me feel slightly better about this. I call it my fan mail. But as you can see, they come just double bagged in those envelopes. And each year we have received between 200 and 300 ticks in the mail. Comparing that to other state programs that are run similarly, that's a lot of ticks. Something about Coloradans being excited to participate in citizen science, and just having a population that's outdoors a lot. We're going to be finding more ticks, even though compared to states east of us, we're a pretty low incidence state for ticks. So in addition to understanding our local ticks more, we are also watching for invasive species through this program. On the submission form, there's a question if the submitter has traveled within the last 2 weeks, where they traveled, um, and when they sent in the tick. This is important because ticks are excellent hitchhikers. When we travel to places in the Midwest, to the Northeast, to the South, where ticks are far more common, they might accidentally come home with us, either on us, on our clothes, on our pets if we traveled with them. So hitchhikers are common. We wouldn't consider that a local detection of an invasive tick. There was one Lone Star tick in 2023, I believe, sent in from Weld County, did not have any travel history. So this was our first chance to mount a field response. So I worked with a group at CDPHE called our field epidemiologists, as well as local epidemiologists and environmental health folk in Weld County. We went out, surveyed the individual's home. They had a small, well manicured backyard, no obvious tick habitats. We checked anyway. We didn't find anything. There was a nearby field as well as a park that had, that tall grass, access to water, wildlife, all the things that are green flags for possible tick habitats. We pretty thoroughly did tick dragging there as well. We didn't find any ticks, let alone any other Lone Star ticks. Um, so the origin of this tick remains a mystery. We have not received any other ticks without travel history from Weld County or any other counties in Colorado for that matter. And these ticks, amblyoma, are more aggressive hunters than other ticks. Um, while a lot of ticks will just hang out on a blade of grass, wait for a host to come by, amblyoma will chase blood meals down. So it is very possible that a neighbor from the submitter traveled outside the state, accidentally brought this tick back with them, and then it crawled into their backyard where it was found. Without surveying the whole neighborhood, we can't be sure. So something new this year, we are incorporating active tick surveillance. So instead of having people send in ticks to us, we are going out into the environment, dragging for ticks in their habitats all throughout the state. This was through that grant that I mentioned earlier, epidemiology and laboratory capacity. And we have over 20 counties participating so far. That's after only one season of pushing this messaging and training health departments, which has been excellent. Another medical entomologist, Cassie Durden, and I have really been pushing this program. And the way that it works is if a county wants to participate, we try to provide them with everything that they need. This includes drag cloths, tweezers for tick collection, collection tubes, sample boxes. They send in the ticks via our lab couriers, so they don't have to pay for shipping. And we've tried really hard to minimize barriers to entry for local health departments that would like to participate in this. Every health department, just like with mosquito surveillance, every health department's doing it a little bit differently. Some are partnering with academic partners. In some cases, it's epidemiologists that want to get out into the field. Sometimes it's environmental health folk at the local health department, or both. Um, yeah, Broomfield is a great example where they are a city county. And so they're able to get not only their epidemiology staff, but also their open spaces, their parks and wildlife folk involved from the beginning. But whatever it looks like on the local level, Cassie and I train them on how to collect ticks. After collection, they'll send those in to CDPHE, where Cassie and I will identify all of the ticks. We'll process them for testing. And then for now, we're sending those ticks on to CDC for pathogen testing. This includes both CDC Fort Collins, where they'll be tested for Colorado tick fever virus, and CDC Atlanta, where they'll be tested for bacterial pathogens. So why do both? Passive surveillance, what we started with, allows you to cast a very wide net at a very low cost. The cost for that program was my time and my microscope. That was it. And that gave us the ability to gather a lot of baseline data to identify trends and then use that information to justify expanding into an active surveillance program. An active surveillance program is much better for actually establishing the distribution of ticks in the environment. In a passive program, we cannot be 100% sure where specifically that tick was picked up, but we can in an active program. CDC also has a strict definition for what is considered an endemic local tick versus an invasive species. You have to detect a certain number of life stages in an area before you can call it tick endemic. So we need active surveillance to confirm that. We also need that for pathogen testing. The ticks that get sent into me in the mail are usually pretty dried up. They are not in great shape. So between missing some of the detailed information on those passively collected ticks and them not being in great shape, that's just not a great use of pathogen testing funds, which is not a cheap process. Actively collected ticks, we can make sure we maintain that cold chain. We can be sure where they came from. That's worth testing. And then lastly, active surveillance is much better at establishing seasonality of ticks, where specifically these ticks are found, what times of year. It also gives us a much stronger baseline of data. With a passive program, if I receive 100 ticks one year and 300 the next year, is it because there are 3 times as many ticks? Or did 3 times as many people hear about the program because of a good news article or because of a social media post that got a lot of attention? That's very hard to determine in a passive program, but in an active program, we can control those variables to get an actual baseline of data. So switching gears again into New World screwworm. New World screwworm, like SLEV, used to be common in the US. Through the release of sterile male flies, we were able to push New World screwworm out of the US, further into Central America, into Panama, where they established a quarantine zone. For a long time, we were able to maintain that boundary. However, a few years ago, that boundary was broken. Ticks were detected further north, and since then, they have been creeping our way. You have likely seen in the news lately, they are now in the US just in the last couple of weeks. They have been detected primarily in Texas, also one case in New Mexico. I say case, these are all livestock detections and one dog. So it has not been detected in a human case in the US since, I believe, the 1960s. Locally, at least, there has been imported cases, I should clarify. Point being, this was a slow creep up through Mexico. We saw this coming. We knew that we had to prepare. So over the last couple of years, CDPHE, meaning myself, other medical partners, our state public health veterinarian, have been working with other partners around the state, the Department of Agriculture, Parks and Wildlife, any agency that might be affected or might be needed in the response to these New World screwworms, we've been working very closely with. That includes running tabletop exercises. We had a great workshop last December where we just ran through different scenarios. All right, what if it's detected in a human that has not left the state? What if it's found in cattle in this part of the state? What about this part of the state? What if the first detection is in wildlife that's much harder to track down and contain? That proved very helpful. And the organizers of that workshop kept emphasizing that they learned a lot from a similar response to high path avian influenza or bird flu or H5N1, where we had dairy, dairy and poultry affected in the state. A lot of the same partners involved. So a lot of lessons learned that can be applied to New World screwworm. Uh, my role in this would be if it shows up in a human in Colorado. If it shows up in a human, that screwworm, that larvae would be sent to me for confirmation of ID before we send it on to either CDC or USDA for further confirmation to be sure what we're looking at. We've also put together health advisories that we are currently sitting on and waiting for if it shows up here. So I want to ask all of you, let me see if I can pull up the chat while I'm presenting. I can, great. Um, I want to ask all of you um, from um, from what I've talked about today, from what you know on either projects that you're involved in or training that you've had, what is the importance of surveillance? Why do we do surveillance? Some of these pictures on the screen might help you. Feel free to toss a hand up, come off mute, drop it in the chat. Um, what's the importance of surveillance of these different vectors that I've talked about today?
Surveillance is knowledge, and knowledge is power.
Yes, absolutely. Um, Power to do a lot of things. That's a good overall statement. But what do we do with that knowledge? Exactly. Anthony says, surveillance enables an effective response to vector-borne diseases before they become a bigger problem. That's one of the most important parts, especially on the mosquito side of things, where we have control tools that we can roll out. We want to make sure that we aren't spraying insecticides unnecessarily. We want those decisions to be data driven. So we want to be looking at surveillance of not only what species and how many mosquitoes there are, but how many of them are infected. Again, each county, each agency approaches that a little differently and what their cutoff is. But I like to see programs that say, okay, when the vector index is at a certain threshold, that's when we go spray and not before then. Some mosquito abatement districts around the US just spray on a schedule. They might spray weekly. They might spray heavily before big weekends like 4th of July coming up when people are going to be outside without that being data driven. We call that spray and pray. And that's not a great approach. That's one of the ways that we end up with insecticide resistance. Then another great example, to have an understanding of the way vectors are changing so we can be prepared for resistance or adaptations in vectors. Excellent. So for this one, we're not only thinking about different vectors showing up, but resistance. Aedes aegypti covers both of those showing up in Grand Junction. When Aedes aegypti showed up, that had to change the way that Grand River did surveillance and control. We're not only thinking about collecting Culex mosquitoes with traps that work well for Culex at the times of day that Culex are active. Now we have to adapt to working on aegypti. And then unfortunately, those aegypti were resistant. So we had to change our control methods as well. Also part of that process was clear communication to the public. They had people hearing that aegypti was in Grand Junction. And, oh, we're all going to get dengue. We had to communicate very clearly that the presence of the vector does not mean presence of the pathogen, especially for something like dengue that is human amplified, not going to be introduced by a bird like West Nile or St. Louis encephalitis virus. It would have to be an infected traveler showing up in the one neighborhood where Aegypti is while they are contagious to the vectors. That's a really good example. What else? Why else would surveillance be important? Well, it's one that we've already touched on. Communication to the public. And not just to the public, but also physicians, veterinarians. Let's say that we do get invasive tick species here in Colorado. Right now, our local species transmit Colorado tick fever virus and Rocky Mountain spotted fever. There are two big concerns. If we had the Lone Star tick, or if we had the black-legged tick showing up in Colorado, um, that changes what we're watching for. We want people to be looking for different symptoms if they're exposed to a tick. We want physicians to be thinking about different things on their differential diagnosis when they see a patient with certain symptoms, as well as veterinarians. Same thing in animals. So I want to wrap up, talking a little bit about that outreach. So something else that is really important in public health is developing messaging tools. Do I work with our communications team at CDPAG to develop tools like those on the screen now? Some that we put out via social media, via press releases, and some that we provide to local health departments, other partners around the state, so that they can message on a more local scale. I also am, as the subject matter expert on these topics, I do a lot of work with the media. That varies a lot year to year. In 2023, I was very busy with interviews, providing information to news organizations that were, very concerned about seeing so much West Nile. This year's also been a fairly busy year in terms of media engagement. A lot of you have probably noticed nationally, ticks are getting a lot more attention. We've had a bad few years for ticks, and this year is worse than it's been in a long time. That on top of starting this new tick surveillance program, we've gotten a lot of very positive media attention about our work. And especially in public health, we will take that positive engagement where that we can get it, especially lately. So this is most, but not even all of the media coverage that we've had this year. For each of these, I provided either data and answers to questions for the media. Some of these also provided an interview for them. And in public health, it's very important that we have those skills to engage the public, not just individuals from the public, but also ways to broaden our message. Sometimes we have to be our own cheerleader. And speaking of media, I wanted to finish with this slide because it's something that bothers me every year, and now it can bother you. Media, they're getting better at this, I swear, but they often use the wrong mosquito when they have a story. It's usually like this example here. This story is about West Nile spiking in a certain area. And that's not a Culex mosquito. That is an Aedes albopictus. Aedes mosquitoes are bolder, they're prettier, they get all of the attention. But it's the wrong mosquito for the story. This bothers me every time. So I want you all to also pay attention to that and also be bothered by it, please. With that, we've got some time for questions. Would love to hear from all of you, questions you have, more about the work that you're doing, anything that we have time for. Thanks, everyone. For questions, feel free to drop them in the chat. You can come off mute, toss a hand up, whatever you're comfortable with.
I have a question. You had mentioned how in 2023 we had a great winter, and that caused a big issue in West Nile. Um, what do you predict the winter this year? Like, kind of the opposite of what 2020.
I That's a good question and one that we get every year. It's a hard question. West Nile virus circulation always starts in birds. Birds introduce the virus. Mosquitoes feed on those birds. Infected mosquitoes then feed on humans. There's a lot of moving parts there, which makes forecasting difficult. And unfortunately, there's not a clear trend of like, oh, it's more wet, there's more mosquitoes, worse West Nile. Studies actually show that drought conditions can actually also cause more West Nile virus transmission. That's because there's fewer water sources during a severe drought, and that drives both the birds and the mosquitoes into closer proximity around those water sources. So you have more mosquitoes infected, or rather, a greater portion of the mosquitoes infected, I should say. That doesn't always translate to more human cases, but there are clear examples from other states where it has. So this year, we also have to think about we had a really mild winter. We had a warm spring. Those mosquitoes, as well as the ticks, they got a head start. Thankfully, those couple of good cold snaps we had in May, I think, notched them back. But we are seeing West Nile detected in mosquitoes earlier than usual. So I'm predicting a average to slightly above average season, and I hope that's as bad as it is.
I saw as one of your headlines in there, Chris, we've already had a West Nile detection in humans. I think that's the earliest ever.
Um, I would have to check if it was the earliest ever. It's really early though. So we did have one human case of West Nile in Jefferson County in May. Um, that's really early. Normally that first case appears sometime late June-ish. So we're talking over a month early. There's an important caveat with that. That individual had traveled to Arizona, where they do have West Nile virus circulating early in the season as well. A lot more West Nile virus activity than we have here right now. But they came back to Colorado right in the middle of their possible exposure window. So it's impossible to determine if they were exposed in Arizona, if they were exposed after they came back. Since they were diagnosed here and they're a Colorado resident, they are being counted as Colorado's case. But I think it's important that we do take it with a little grain of salt. Abby, that's a tough question. So Abby asks, from my experience, how do public health agencies adapt projects in response to funding cuts? I am very lucky that I don't have too much experience with this. As certain public health topics have become more political and have experienced funding cuts, as COVID funding has wrapped up and those teams lose people, those have not really affected those of us in zoonoses and One Health sectors. Um, Everyone loves bugs and loves to hate the bad bugs. So I haven't been directly affected by that. But looking at the rest of the health department, um, we do what we can with what we have. As COVID funding has kind of wound down, we have done all we can to keep epidemiologists that have been through that, been through the ringer. That is about as good of training as you can get in epidemiology was the COVID pandemic. To keep them in-house. And that might have been moving teams, that might have been changing their classification. A lot of ways that we have tried to adapt. And I'd say that I'm not strongly affected. That's with the important caveat that my work is very dependent on what local health departments can do. Local health departments that are experiencing those cuts might say, we need to cut back on West Nile surveillance this year. Our environmental health team needs to focus on restaurant inspections, water quality, pollution, other things that they feel are more important. Or maybe they feel like they don't have the capacity to take on a new tick surveillance project. In that way, I have been affected by this, but not significantly yet. We'll see what the future holds. For Anthony, question, how do you compensate for underreporting from hospitals and communities? That very much depends on the pathogen, the vector, whatever the reporting might be. A good example is West Nile virus versus St. Louis encephalitis virus. West Nile virus in Colorado is what's called a reportable condition. If someone is diagnosed with West Nile, that case has to be reported to the state. As such, we are not missing any confirmed cases of West Nile virus in Colorado. Confirmed is an important qualifier there because we know about 80% of West Nile virus cases are asymptomatic or have very mild symptoms, so they're not showing up to the hospital. They're not getting diagnosed. We just have that in the back of our minds that if we have, 300 cases of West Nile, it's a lot more out there that we're not seeing because they're not reporting to the hospital. They aren't in the reported cases. Other diseases, Colorado tick fever virus, for example, is not reportable. So in order to get a good picture of how many cases there are around the state, sometimes we have to search hospital records. We have to look for that case data in order to get our information. Did that answer your question, Anthony, or is there something else you were curious about? Awesome. Well, as we wait for possible more questions to come up, my email is on the screen. If any of you are curious, want to get nerdy about certain topics, have questions about things that you're working on through RaHP VEC, or any other questions about working in public health entomology, don't hesitate to reach out. I enjoy chatting, so would be happy to.
Yeah. Feel free to send Chris some ticks as well. I guess he'll need a
Please
little bit,
do,
but we can get that to you.
especially those of you out doing field work this year. If you encounter a tick, send it my way.
Yeah. And if you are interested in getting involved in some of the active tick drags, let me know, and I will try to get you in touch with your local people that are doing it. We'll see if we can come up with something. So it's a lot of fun. I wish that I had Chris doing this program when I was in school so I could go out and hike and find ticks. So we got another question.
Question from Nolan. Do I have general advice for people interested in a career in public health or medical entomology? So I'm going to plug another one of these seminars coming up, I believe, next month, where we'll have myself, someone from academia, and someone from an actual mosquito control district on more of a career panel, talking about our paths into our careers and taking these kinds of questions. But while you're asking this question now, two skill sets that you cannot ignore if you want to go into this field. Vector identification will always be core to a medical entomologist's role. Going through school, I was mostly a mosquito guy, but I made sure to get broad training where I could get it. I learned the basics of tick ID. Those basics allowed me to significantly expand our tick surveillance program. If I hadn't made sure that I got that training, it would have been a lot harder for me to learn after the fact. And another one is data. I should have gotten more training and experience on data analysis and management when I was in graduate school. I got some of the basics, but I didn't go out of my way to seek out more, and I wish I would have. Um, a good example is during my PhD. I started my PhD program in 2014, um, and within a couple of years, Zika had hit South America. Um, I remember a discussion I had with my advisor. I had a totally different dissertation mapped out. And at one point, we were chatting in his office, and he said he had a collaborator in Brazil who wanted to do more field work, more mosquito collection, more insectary work for Zika, and asked if I would be interested and if I wanted to spend time in Brazil. Said, absolutely, let's trash my dissertation plan and we'll just go in this direction because this sounds way cooler. But because of Zika, everything had to be published in a hurry. We would finish an experiment and the data analysis had to be done yesterday. Because of that, a lot of that data work got farmed out to collaborators who had the skills, who were very good at it, who could turn it around overnight. And I got a little shortchanged along the way. It was exciting to be part of, such a fast-moving field, but I didn't have to struggle through that data work and develop those skills that honestly I'm having to do a lot more in my current role, taking more training in how to use R for environmental studies, learning new ways to do productive data management for successful analyses, things like that. Yeah, so those two skills, along with and another one that I've learned is kind of rolling with the punches. Zika's a good example of that. A broad skill base and flexibility allows you to take advantage of projects. My postdoc got taken over by COVID. During my PhD, I was the mosquito guy in an arbovirology lab. During my postdoc, I was the arbovirology guy now in a vector lab. Then COVID hit. Our studies totally changed. We ended up studying COVID transmission in household pets in homes of human cases. And instead of doing field work, I was in the BSL-3 running viral samples and PRNTs. Not what I wanted to be doing, but that's where the funding was, and it allowed us to, take advantage of the field. Oh, one more piece of advice that's on my mind. I had a very clear path through school. I went undergrad, master's degree, PhD, postdoc, current position. I was lucky. That was great. I know a lot of people that enter the workforce either from undergrad or after getting a master's degree, either an MS or an MPH, going into the field. You don't always need a doctorate to be working at this level. Most of the people I work with at the state health department are an MPH or an MS of some sort. Some of them along the way, after they're in that position, seek out further degrees. That's a great way to do it, a great way to get into the field. Make sure you know that you enjoy it, and then identify how an additional degree will further advance your role.
I'll just add a little anecdote as far as the flexibility thing goes. And a RaHP VEC success story of sorts, I guess. It's unfortunate none of them are on the call today. They're all out in the field right now, but we have some interns down in Albuquerque, and last year using a collaboration with a professor at the University of New Mexico, with some old COVID funding, we were able to institute some West Nile testing and SLEV, I think, actually, now that I think about it, testing down in Albuquerque. So not necessarily a career flexibility, but that application of different funding to benefit us. And then we were able to use the data that they generated they were able to use the data that they generated to ask the state for dedicated funding, which they received over this year and next year. And hopefully it's a little bit of a spiral, but hopefully that pilot funding that they got from the state will allow them to then create a permanent program from there. And so sometimes these things can kind of snowball, and yeah, so just wanted to kind of highlight that. So we are at the hour. If you have any more questions, I'm sure Chris would be happy to answer them. And like you can see here, his contact information is there. You can always reach out to me, and I will just share it again when I send out this recording to everybody, just in case you think of something in the next couple days or whatever. You're more than welcome to reach out. So I am going to end it there, and thank you all for joining.